Hello everyone, so today we'll be having a short lecture. So we'll have a short series of Immunolab lectures looking at different techniques used in immunology, probably the more common ones, but today we'll be looking at hybridomas and monoclonal antibodies, which you've seen in our immunotherapy lecture if you want to take a look. So polyclonal versus monoclonal. So polyclonal antibodies are a collection of antibodies specific for several different antigens or epitopes. These are usually obtained from the serum after immunization with an antigen of interest. Where monoclonal antibodies, which are all are specific for one epitope. So these are produced by a clone of cultured B cells, making one antibody specificity only. So we want to find one receptor, say we're just looking for CD3 only. We have a monoclonal antibody specific for CD3, will exclusively bind to CD3. Or even more specific than that, it would be the epitope on CD3 that we want to find. So how are these monoclonal antibodies obtained? So basically this is made through hybridomas. So hybridomas are a product of cell fusion between spleen B cells from an immunized animal and myeloma cells, which are cancerous B cells. So these cancerous B cells, like many cancer cells, proliferate uncontrollably, and these continuously and they proliferate continuously and produce antibody. So essentially, we have a cancer cell that keeps dividing, but we have it where these are specific B cells that produce a certain antibody of interest. So this antibody binds to IL-8, which I'll be using in my thesis. So essentially, these hybridomas can only produce this one antibody since it's this one singular cell being divided over and over again, and we can just constantly have these cytokines being produced. And essentially, if they're kept in proper lab conditions, they can essentially live forever because they're cancer cells. We're allowing them to divide uncontrollably. And this process was developed by Kohler and Malstein, and they won a Nobel Prize in Physiology for this. So this was a huge advance in the field, and also allowed us to study antibodies because we essentially had consistency in knowing exactly what we're looking at, and we could break apart the antibodies and look at them in detail, and we could basically have an unlimited supply to study without having to take them from someone's tissue or something like that. So here are the key steps in making these hybridomas for monoclonal antibody production. So first, we immunize a mouse with an antigen of interest. So like again, anything we can think of. There's so many different options. You can go to certain websites. I think there's R&D systems and a bunch of different biotech companies produce a ton of these for labs and researchers and like huge pharmaceutical companies. So after this mouse is immunized, this mouse responds to the antigen and B cells are stimulated to produce antibody. The spleen contains many antibody-producing cells, and then we remove the spleen and fuse B cells with myeloma cells. This occurs in a polyethylene glycol, which causes cell membrane fusion. These successfully fused B cells and myeloma cells form the hybridoma cells. So now we culture it and test for antibody production, and we need to screen for hybridomas producing antibody and make sure it is specific to the antigen of interest. Essentially, these are cultured, and here's a good figure to summarize it. So again, our mouse is immunized with antigen X, which can be anything. Spleen lymphocytes are removed from the mouse. Myeloma cells are in tissue culture. They're mixed to make the hybridomas. Hybridoma grows in tissue culture. We select the hybridomas that make antibody for that antigen. The rest are removed. Then we have individual cells are cloned, and then our antibodies to antigen X are present, and the specimen is saved, and then they keep dividing, we keep taking them out and out and out, again and again, and then we have these in bulk. So we use a specialized media, and myeloma cells are used during monoclonal antibody production. So HGPRT is hypoxanthine guanine phosphoribosyltransferase, so that's a long one. HGPRT is slightly easier, and this is an enzyme required for the uptake of hypoxanthine and thymidine from the media. So HGPRT deficient myeloma cells have a lack of HGPRT, results in the inability to survive in selective HAT medium. And HAT is hypoxanthine aminopterin thymidine. So aminopterin blocks de novo nucleotide synthesis. These mouse B cells do have HGPRT, so they can take up hypoxanthine and thymidine from the medium, so they would survive, and they survive for a while. So again, we have our specialized medium, you can see here, and again, mouses do have HGPRT. So then there's a selection process. So myeloma cells die due to the lack of HGPRT. Spleen cells die and can only undergo limited number of divisions in vitro. So the sole survivors are only the successful fused hybridoma cells that survive in HAT medium. So essentially, the only ones that survive are the ones that will proliferate and survive. So here are spleen cells producing antibody from mouse immunized with antigen. Our myeloma cells are immortal cells lacking antibody secretion and the enzyme HGPRT. You mix and fuse the cells. Then we have the transfer of the HAT medium to HAT medium, and these immortal hybridomas proliferate. And the spleen cells and unfused HGPRT myeloma cells die. So again, the sole survivors are the hybridoma cells. So the desired product of this reaction is a hybridoma cells producing identical antibodies specific for the antigen of interest, which is the monoclonal antibody. See, so this is continuing the figure we just saw. So now we have select hybridoma that makes antibodies specific for the antigen. So here it is. Now this will be cloned, and we will now clone the selected hybridoma. And again, this is our specific antigen, and this can either be specific to the certain cell or even the epitope. So mouse spleen B cells do have HGPRT, and this makes antibody. These myeloma cells do not have HGPRT and are not making antibody. But myeloma cells are immortal, and then we have our selection process. So myeloma cells die but cannot make nucleotides. If they cannot make nucleotides, spleen cells die due to their limited number in vitro. And once we have the successfully fused hybridoma cells, which have properties of both, they possess HGPRT from the B cells, they are immortal from the myeloma property, and ideally they are making the antibody you want. Again, this can be summarized in this figure here. So the production of monoclonal antibodies, so only the hybridoma cells grown in culture will survive, like we said. Any unfused spleen cells cannot divide, and any unfused mouse myeloma cells cannot get the nutrients they need to grow. So again, here's a huge summary figure. Essentially describes the whole process. We already went through this, but I recommend taking a look again. So the next steps. How do you detect the desired antibody producing hybridomas? So we carry out limiting dilution process, where each hybridoma cell in an individual well, we put each individual cell, so this would be the well. You can see they're burning it in here. And these allows them to grow and secrete their antibody. Then we screen the culture medium for the presence of the desired antibodies we want. Then we use our enzyme immunoassay or immunofluorescent approach, so our ELISAs and several other techniques. Next, we expand this culture of selected hybridoma cells producing the desired antibody. Then we can have large-scale monoclonal antibody production. So we can grow in large volumes in tissue culture systems and collect monoclonal antibody-rich mediums, and can also inject mice in their peritoneal cavity and have ascites, which is a fluid-rich, causing ascites, which is a fluid-rich in monoclonal antibodies. So essentially, so a lot of these are used in medications, so they actually have like massive scale production of these antibodies. And again, those cells are immortal, so we produce tons and tons and tons and tons of antibody, and these are put into different medications, and they're also used to treat certain conditions, like straight up you've probably heard about in COVID. There's some people where they took antibodies at previous people who were exposed to infection, who had immunity. They would inject those once they're screened for safety and removed from the blood into other people. So yeah, they have a lot of therapeutic effect, and it was a huge, like this whole discovery and the way they produce these essentially changed immunology and immunotherapy. So again, why make monoclonal antibodies? So they're extremely important in diagnostics. Since we can make them specific for even epitopes, we can tag, not only, they don't have to act as antibodies in the sense of destroying things, they can tag different receptors. So again, CD3, This, for example, we can tag this with a bunch of antibodies, and we tag certain dyes to them, or certain molecules that will light up when they detect them. So here you can see how they colored, and then we can essentially count how many there are, and it allows us to detect with detection of both pathogens and toxins in clinical samples. So CD3 is not a toxin, but just another example. They can be used in research in detecting the expression of proteins of interest on cell surfaces like CD3 in tissues and blood and cell culture products. And they're highly used in immunotherapy. For example, cancer therapy, which we can attach a toxin to a monoclonal antibody specific for tumor cells, and this is targeted delivery of the toxin. We can use monoclonal antibodies for the detection of specific antigens by microscopy. So on the left here, we use immunogold electron microscopy, where antibodies coupled to the gold particles used to identify Enterococcus facilis. And here on the right, we have a fluorescent labeled monoclonal antibodies used to detect an ion channel protein in the epithelium. So again, this helped with so many scientific discoveries, and as well many immunotherapeutics. So applications. They can help with identification of cells expressing certain surface molecules. So we can use antibodies specific for certain cell surface markers to identify cell types of interest in a mixture of cells. So we have fluorescently labeled monoclonal antibodies, which can be used to identify cells of interest using the process of fluorescent activated cell sorting, or FACS. Specific cell types can be physically separated from a complex mixture of different types. So we can put in a bunch of different, here we can see the color molecule, fluorescent positive cells, we can put in a bunch of different antibodies as well. So one can be green, so we can have one green, one yellow, for example, and then we can have, these will detect different markers. So when we go to count, we can know, okay, the green was the macrophagy and the yellow was the NK cell, for example. So it allows us to kind of see what stuff is happening, and it's pretty impressive. So here we can see we add antibody that recognizes lineage-specific markers labeled with a red fluorescent dye. Then we have fluorescent activated cell sorting, and negative cells and positive cells. And then these can be detected using ELISAs or flow cytometry and other stuff like that. So another therapeutic application is immunotoxins. So monoclonal antibodies can be fused with bacterial exotoxins or toxic agents. This toxin will block the protein synthesis in the target cell. So A to B toxins. So toxin B subunit binds to a cell. This is internalized. A subunit has a toxin activity, for example, Shiga toxin, where the A subunit prevents ribosomal subunit assembly process. So this is kind of summed up in this figure. So again, immunotoxins, the B subunit of a toxin is replaced with monoclonal antibody specific for the tumor cell surface molecule, and this binds specifically to the tumor cells and delivers the toxin. So again, it's a targeted attack. So there's some issues with mouse monoclonal antibodies. So for example, in immunotherapy, so in cancer therapy, we attach a toxin to a mAb specific for tumor cells, and again, this is targeted delivery of the toxin. The problem is that humans respond to mouse antibodies as foreign. And so we have a human anti-murine antibody response, or HAMA. So solutions to this are to use chimeric mouse-human antibodies. So this is when we're essentially merging mouse and human. So our constant regions, human, and then our variable regions is mouse. So the reason we can use mouse here, the reason why we do it is that we can generate those myeloma cells and basically get any specificity we want. But this will give us that IgG or M, etc. But usually we use IgG. But that's a human form. So that's the thing that isn't as variable, like we talked about in our antibody lecture. That this is the part that is essentially different between the different antibodies. So all IgM molecules will kind of have the same constant region. Again, hence the word constant. So we can change that variable region, and this still counts. Our body recognizes it still as self when we use chimeric. We can also use humanized antibodies, which have both a human constant region and a human and mouse variable region. So in this case, the human variable framework region, we have a variable framework region, and then the mouse hypervariable or CD R regions. So the advantages of this is that monoclonal antibodies have the desired target specificity, but are less likely to generate anti-mouse human responses in human recipients in these two solutions. So again, a lot of crazy advanced science going on here. Whoops. So I'm not going to read this all to you because it's a lot, but this is a great thing if you want to just check out on Nature is the milestones in antibody research. There's also milestones in vaccines, which I also recommend and we use for our lectures. Essentially, it goes through the huge discoveries in antibody research. And again, some of these are really, really impressive and changing the field essentially. And again, it's only going to get better, especially with SARS research coming out now. So there's huge funding into immunology research, which is great. In an unfortunate circumstance, but we'll likely have a lot of great discoveries coming soon. Hope you enjoyed it. Hope you learned something. I'll see you in the next one.